All right, so now that we know a little bit about some of the basic properties of carbohydrates, we can turn back to what we really want to understand, which is this idea of extracting energy from these structures, right? And so that leads us to the discussion of this first fundamental pathway, which is the catabolic breakdown of glucose, right? And so I guess a nice place to start would be, well, why glucose? I mean, I just sort of explained there's pretty vast space here in terms of the diastereomers of these sugars. Why glucose? Well, one of the reasons one of the ways in which to answer that question is nature, as we've talked about, tends to like symmetries when it can get to symmetries, and the process we're going to see is going to break the sugar into 2 3-carbon units, and that symmetric chemistry kind of requires at least a 6-carbon sugar. Could do it with a 4-carbon sugar, but then you're not going to get very much out of it. Using odd numbered sugars requires a lot more steps, as we would see if we had time to study the pentose phosphate pathway, which we're not going to get to. And once you get up to 8-carbon, I mean, just for whatever reason, biology just decided that's too much trouble. And so this 6-carbon aldehexose is the sugar that is the starting point. Now, why not one of the other diastereomers rather than glucose? Who knows? This may be another one of those examples like D versus L amino acids that for whatever reason, there was a excess of D-glucose around abiotically for some reason, I don't know. And evolution just got started and locked onto it, right? But the observation is that glucose is a very common, perhaps the most common, and in many cases, the preferred fuel that's going to go down this pathway that we're going to be talking about. And for some cells, it's the only fuel they will use. For example, red blood cells, brain and central nervous tissue, some of your other tissues will, at least CNS, except under stress conditions or non-starvation conditions, will only wants to use glucose. Red blood cells will only run on glucose. Some other tissues will only run on glucose. So it is an absolute requirement for many tissues in organisms like us that glucose be available as a fuel to power this particular pathway. Again, the particular pathway, of course, as you hopefully remember from your undergraduate biochemistry, is of course glycolysis, which fundamentally, or quite literally, means glucose splitting, right? Which is what we're going to see. The glycolytic pathway involves cutting glucose essentially in half, right? Okay, and this is a universal, it's an ancient pathway. It's essential to essentially every cell-based living organism on this planet, as far as I know. It's probably one of the very first metabolic pathways to evolve on this planet. It certainly predates the oxygen-saturated atmosphere, which we currently enjoy and which we evolved in. And in fact, the energy balance of broader metabolism in organisms like us shifts dramatically in tissues when oxygen is and is not available, right? When under so-called anaerobic conditions, the energy balance of these pathways changes fundamentally, and the metabolic output redirects in a fundamental and important way, as you're going to see. Glycolysis is sort of, you can think about it as the fundamental ignition switch sitting at the top of intermediary metabolism. That's really critical for getting not only energy production going when one has sugars available, and also for other important types of inputs. But the chemical transformations in the intermediates that this pathway creates are fundamentally important for doing all kinds of other types of important biochemistry, principally many aspects of biosynthesis and anabolism depend upon running this pathway to create intermediates that are necessary for making other stuff. So it's important not just for energy production, but for also for anabolism and for making stuff. Now, the study of the glycolytic pathway, as I mentioned before, started really in a big way in the 1930s, in sort of the detailed way where this pathway was really worked out by some German chemists by name Emden, Meyerhoff, and Warburg, right? In fact, some people you may hear refer to glycolysis as the Embden-Meyerhof pathway. That was an old way it was referred to, and you'll still hear people name drop that. But if someone is being fancy and saying the Embden-Meyerhof pathway, they really basically mean glycolysis, right? And the overall process that we're going to be talking about, of course, is, like I said, this glucose splitting reaction, where we're going to take that aldohexose glucose, right, that I've drawn out for you guys several times, right? Right. And we're going to basically split it into 2 3-carbon units of this simple carbonyl containing carboxylic acid compound, pyruvate. Oh, come on. Right. This is the fundamental goal of what we're going to be doing with glycolysis, is turn glucose into pyruvate, and in the process, pull out some electrons and make some ATP, right? That is our big goal, right? All right, but to do this, we're going to have to go through some fundamental steps, right? So, whoops. So here again is just that big overview, what we're going to be talking about. We're going to take these monosaccharides, specifically glucose in our case, and we're going to come down here through this the first part of the central trunk of metabolism, glycolysis, to ultimately make pyruvate, these 2 3-carbon derivatives of a glucose that's been cut in half, right? And you can think about glycolysis as having sort of 2 phases, right? This helps me remember it anyway. The first few reactions of the glycolytic pathway, you can think of as the so-called energy investment phase. We got to put some energy in to get this reaction to go, because we got to prime, kind of think of like prime the pump. That glucose carbohydrate is relatively stable all by itself, right? It's not going to fall apart on its own. And to get this molecule sort of prepped for this splitting reaction to break the bond between that carbon-3 and carbon-4, we really kind of have to destabilize it a little bit. And that's going to involve burning a little ATP to set the thing up to go. Once that's happened, right? Once we've essentially phosphorylated this glucose molecule, the pathway then becomes fully exergonic, fully exothermic, and generates this reasonable output of both synthesized ATP and some high potential electrons, right? And to do this, to run this reaction, or run this conversion, we're going to have to go through about 10 steps, right? So the conversion from glucose down here to pyruvate is going to take about 10 reaction steps to get this to go, right? Separated, as I said, in this sort of energy investment phase and this energy generation phase. All right, and in the next video, we're going to dig in to the beginning part of this energy investment phase and see how this all gets set up and gets us down to the point where the pathway then, once we're down here, becomes a fully energy producing pathway.